Cellular respiration, specifically glycolysis and the formation of acetyl CoA. Every living cell in the human body is in need of an energy source to keep them alive and functioning. The cellular respiration process is a key component in allowing cells to maintain a constant internal environment. The overall goal is to make ATP. The first step in cellular respiration is called glycolysis, which takes place in the cytoplasm of the cell. We have our glucose molecule, which is composed of covalent bonds or sharing of electrons. The splitting of these covalent bonds causes an exertion of energy. The energy that is let out can be used for the formation of the cell's energy source, or ATP. The next step is to isolate the 6-carbon chain from H2 and O6. The process of removing the 2-chain hydrogen from the molecule requires the use of the coenzyme NAD and oxygen obtained from breathing. One of the main functions of oxygen is to break apart the hydrogen from the glucose molecule, turn it into water, and create ATP. Keep in mind, when the hydrogen bond is removed from the molecule, we call it oxidation. We are now going to focus in on splitting our 6-carbon chain of glucose. A phosphate group is added to the 6-carbon chain by using the energy source ATP. This can be referred to as the investment stage because we use up ATP instead of forming it. When phosphate connects to the carbon chain, it produces glucose 6-phosphate. Isomerization, which is defined as changing one cluster of atoms into a new molecule simply by changing the sequence of atoms, will occur. Our glucose 6-phosphate will become fructose 6-phosphate. With the use of additional energy, another phosphate is attached to our fructose 6-phosphate, leaving us with a 6-carbon chain of 2 phosphates attached called fructose 1,6-biphosphate. Next, our isomerized molecule gets divided right in half, forming dihydroxytone, commonly referred to as DHAP, and glyceraldehyde 3-phosphate. Isomerization once again takes place and changes the GHAP molecule into glyceraldehyde 3-phosphate. Our goal in this process so far is to get to the stage of making pyruvic acid. In order to get there, hydrogen ions, or H+, are added to the coenzyme NAD+, in order to make NADH. After this occurs, we are left with 2 biphosphoglyserates, 1,3, which has 2 phosphates attached to the molecule. We have 2 of them. Next, biphosphoglycerate 1,3 is catalyzed by phosphoglycerate kinase and transformed into 3-phosphoglycerate. A phosphate is removed from the biphosphoglycerate 1,3 in the process and is given ADP to make ATP. Let's take a look at how many ATPs we use in this process. Our 2 biphosphoglycerate 1,3s made 2 ATPs. But if we look back in the investment stage, when we broke down our 6-carbon molecule, we realized that 2 ATPs were used in that process. We now have a net total of 0 ATP. Looking back at our 3 phosphoglycerates made from biphosphoglycerate, we are now going through the process of isomerization once again. 3 phosphoglycerates will get changed into 2 phosphoglycerates. The enzyme elocase speeds up the next step, which involves taking away the H2O from the 2-phosphoglycerate. When water is taken away, phosphopyruvate is created. In this reaction, phosphate on the phosphoglyceride is prepping to let go of the phosphate to be used in the next step. Finally, phosphopyruvic acid exerts its phosphate in order to turn ADP into ATP. This is all done through a process named substrate level phosphorylation. The next step in the process is called oxidative decarboxylation of pyruvic acid into acetyl CoA. It's very important to form acetyl CoA because it's a key component for the citric acid cycle. Dehydrogenase multi-enzyme assists in the process. The first stage in this formation is taking place in the mitochondria where pyruvate is sent. The catalyzer pyruvate dehydrogenase has one duty, and that is to take off the CO2 molecule from a single pyruvic acid. TPP assists in taking away CO2 and connecting itself with pyruvic acid. Lipoamide is added to the group and TPP is taken away. When this happens, we are left with acetyl. Next, lipamide gains oxygen, which makes our coenzyme A. Coenzyme A attaches itself to acetyl, creating our end product of acetyl CoA.